Hey, here's something wild. Many of the biggest advancements we're seeing in biotech right now are the direct result of a several billion years long arms race between bacteria and the predatory phages that hijack their cellular machinery. For real, one of the biggest scientific breakthroughs this century was this class of cellular machinery. This is called CRISPR, a bacterial countermeasure against the viral horde that can break invading DNA before it causes havoc. In the last decade, we've repurposed this CRISPR variant, CRISPR-Cas9, as an incredibly powerful and efficient tool for gene editing. But I always loved the metaphor that CRISPR was the result of an arms race with viruses. And it turns out that metaphor is a pretty solid way of thinking about things, because in the last handful of years, we've actually isolated and refined anti-CRISPR proteins to disarm the bacterial defense systems, giving viruses a leg up in this endless evolutionary wrestling match. A new one of these anti-CRISPR proteins was just isolated and research published earlier this month. This one might be a pretty potent way to disarm Cas13b, the cool, edgy RNA slicing cousin of the CRISPR-Cas9 system you've probably heard of. And honestly, there's a fire hose of amazing research hitting the Biochem newswires lately. I think this is one of the most important moments in the history of biology to try and be dialed into this industry. So I'm not going to waste any time here. Let's explore two of the coolest papers I saw this month and see what we can learn about this present moment in biochemistry. We'll meet the latest anti-CRISPR on the block and check out a powerful new breakthrough that will help scientists unlock some of the biggest secrets in a kind of cell division called meiosis. There's a lot to cover, so let's not waste any time. Let's meet some of the latest and greatest papers in molecular biology. And hey, welcome back to Clockwork, a YouTube channel about biochemistry. Full disclosure, this video is being sponsored by Brilliant, but we'll hear from them a little later. Usually this channel is focused on in-depth videos that fully illustrate a single large topic in molecular biology. But I'm trying a new segment today where I bring you a handful of surface-level stories covering freshly published research. There's so much incredible data getting published across the life sciences every week, and I want to take a shot at covering it as it comes out. Subscribe here if you want to see a monthly breakdown of my favorite snapshots in biology, or head over to watchclockwork.com and sign up for my newsletter if you want those little takes, a little bit more often and a little bit more like in written form. But for now, let's get to the clickbait immediately so I don't test your patience too hard. And yeah, the first paper we're covering is a real doozy. Let's check out the latest discovery in academia's long history of uncovering the ancient arms race between bacteria and the viruses that constantly haunt them. Let's meet ACR13b and see how it totally hamstrings anti-RNA CRISPR proteins. And so yeah, this one's kind of a hard one to start with because we're at the cutting edge of cutting edge research. If you're even reasonably plugged into recent developments in the life sciences, the title alone here is going to be wild. And I mean, y'all have heard of CRISPR, right? It's a bacterial defense system that's been re-engineered by human scientists for the purposes of gene editing. While I haven't had a chance to cover CRISPR in my core Central Dogma series, and don't worry, I'm definitely getting to it later on, we can still do a super fast primer right here before we talk about what an anti-CRISPR system is. And again, I'm boiling this way down to the absolute surface level here. So the CRISPR that you've probably maybe heard about is actually CRISPR-Cas9. Turns out there's actually a lot of CRISPR systems. Now, Cas9 is a class 2, type 2 CRISPR complex. That's right, it turns out there's two major classes and like half a dozen types of CRISPR machines that scientists have discovered since Jennifer Doudna and her team isolated Cas9 back in 2012. Cas9 works by using an RNA template to find and cut a specific DNA segment, which is what makes it awesome as a genetic engineering tool. We've since developed CRISPR treatments that can cut out DNA responsible for all sorts of genetic maladies, and the horizon for tech like this is only getting broader as folks in the industry develop more sophisticated protocols. Bacteria evolved systems like Cas9 to quote-unquote remember the DNA from viruses that attacked their community in the past. It's the closest thing to an immune system that a single cell can hope for. A DNA-based bacteriophage injects its DNA into a target bacteria, and this Cas9 system is going to be holding on to a small piece of RNA that acts as memory of that specific DNA sequence, which will then help the protein side of this Cas9 system to recognize and cut that viral DNA before it can hijack bacterial protein factories to create more viruses. Nice. Here's the thing though, not all viruses use DNA to infect their hosts. A big portion of viruses are actually RNA-based, cutting out the middleman entirely and injecting mRNA to speed up their infection process. But there are CRISPR systems purpose-built for dealing with that, which finally brings us to the subject of our current paper, CRISPR-Cas13b. a Class II Type 6 system built around targeting and destroying RNA-based attacks. Scientists have uncovered a lot about Cas13b in the past few years, and we've even engineered versions of it that are showing promise as RNA editors and antiviral treatments. Why go through all the trouble and drama of editing things like heritable DNA when we can just continuously modify problematic RNA transcripts? And here's the thing though, again, we've described these CRISPR systems as the result of a multi-billion-year arms race between bacteria and these viral predators. And again, that's honestly a great metaphor because now we're starting to discover anti-CRISPR systems. That's right, viruses and other genetic parasites are incredibly crafty. It turns out that a billion years of selective pressure is enough for viruses to evolve systems that counteract bacterial CRISPR defenses. And this paper discusses ACR6B1, a gnarly little anti-CRISPR protein that potentially has a very clever method of blocking Cas13b from doing its job. Let's check it out. So first up, the team behind this paper did an awesome job isolating a crystal that allowed them to render a solid model of Cas13b. This RNA slasher has two main parts. This little pocket up here binds to the main CRISPR RNA loop, which is attached to the spacer RNA that the CRISPR uses to identify and destroy viral RNA. Basically, this single RNA strand is complementary to a viral RNA our hero bacteria remembers from a previous infection that hit it, or more likely, a community member. If that same viral RNA gets injected into our bacteria here, Cas13b will automatically bind to the complementary sequence, and using these two HEPN modules down here, it'll slice the foreign RNA, rendering it useless. And that's really cool, but this paper has illustrated how a virus-encoded protein completely blocks this process. And so, meet ACR6B1, anti-CRISPR protein 6B1, or the first confirmed anti-CRISPR protein that inhibits a type 6b CRISPR system. And I mean, come on folks, the acronyms in emerging life sciences research are half the fun here. Now, what you're looking at may not be the actual structure of A6B1. This is the structure as determined by the AlphaFold 3 server. The researchers who wrote this paper did get a low-resolution cryo-EM map of A6B1 binding to and changing the shape of Cas13b, but it wasn't high-res enough to fully confirm their proposed model. So what you're about to see is a simulation of the mechanism that this microscopy data suggests. Basically, Cas13b changes its shape a lot when binding and processing its CRISPR RNA. This segment up top potentially acts as like a hinge that opens up and closes to allow the CRISPR RNA to bind to the pocket right here. Once the RNA is loaded, this hinge will close and help activate the cutting part of Cas13b. Unless, of course, ACR6B1 is in the system. Check this out. Our anti-CRISPR protein will act as a monkey wrench thrown into this system, binding to this lighter helical region during that moment where the CRISPR RNA is being loaded. This keeps the scissors down here from properly activating, and also potentially holds the target RNA outside of the system, kind of rendering it impossible for it to use. This jammed Cas13b can't do anything really, so our viral mRNA is free to bind to a ribosome and then turn this bacteria's internal machinery against itself. Diabolical. There's still a lot we need to learn about Cas13b in order to verify the mechanism and conformation that this cryo-EM map suggests, but this paper represents another massive step forward in our understanding of the broader CRISPR arsenal. While Cas9-based gene editing systems occupy most of the popular science reporting, with approved treatments for sickle cell disease and a handful of other genetic disorders, along with fears about engineering designer babies and stuff, RNA-first systems like Cas13b may have way more interesting applications as we refine our understanding of this antiviral weapon. A refined Cas13b may be promising as a straight-up antiviral treatment for diseases like COVID-19, where deploying a variant of Cas13b loaded with a COVID mRNA sequence can destroy the viral infection at the very second it starts. However, there's a lot of concerns about how specific Cas13b can be in a system as dynamic and complex as a living person undergoing an immune response. Having a potential off switch in the form of an engineered version of ACR6B1 on deck could be a huge addition to any potential treatment protocols that are currently being developed. But I really don't want to use this channel to get too speculative when the science I'm writing about is this fresh. For me, this paper is a huge deal because of how this team got these results. This is actually a follow-up to a previous paper this team put out a few years back. In that paper, the team threw a bunch of bacterial and viral genomes at a machine learning algorithm to try and find any sequences that might code for anti-CRISPR proteins. ACR6B1 was the biggest hit that computer model came up with. So this paper took that ACR6B1 sequence, synthesized that protein, and then tested how it would interact with Cas13b proteins in vitro. Going from computer model to real proteins pure enough to be resolved in electron microscopy actually this fast, is unfathomable. As someone who basically writes for a living, I'm not super sold on the more popular narratives about AI right now. While AI agents are great for making infrastructure tasks much faster, chatbots aren't remotely ready to take over most human writing. However, in the life sciences, AI is shattering barriers to discovery by allowing researchers to speed up their discovery process. There are limitations still, obviously, but thanks to AI, we are currently experiencing what feels like a significant acceleration of our ability to understand the base mechanics of living systems. Because remember, sometimes it's more important to have a coherent model of how a mechanism could work instead of trying to isolate that mechanism in the cellular pandemonium of a fully living thing. And you know what, speaking of models, after 25 years, it looks like scientists have cracked a way to better research one of the most important single moments in all of cell biology. So you know what, let's pivot up to Eukaryote Town and talk about finally having an in vitro model for one of the very first events in meiosis. But of course, before we do that, let's check in with the sponsor of today's video. Today's video is brought to you by Brilliant, where you learn by doing. The most important way I learn is by incrementing my way through a topic. Even something as immense as gene editing via CRISPR is learnable as long as you tackle it in reasonably bite-sized chunks. That's what makes Brilliant great. It's a learning service with in-depth and incredible lessons that can gradually level up your understanding of the harder sciences. Honestly, relearning math fundamentals, now that they have these really great visual algebra lessons, it's a great way to sort of consistently develop the math side of my brain. Each lesson is filled with hands-on problem solving that actually let you play with concepts, which is a method proven to be 6 times more effective than watching lecture videos. All content on Brilliant is crafted by an award-winning team of teachers, researchers, and professionals from MIT, Caltech, Duke, Microsoft, Google, and so many more top-tier institutions. So while you're establishing a real basis of real knowledge on specific topics, it also helps you work those cognitive processes to help you become a better thinker. More importantly, Brilliant is an incremental platform which will help you develop a powerful daily learning habit. You're not going to learn algebra or data science or calculus in a single day, but you can incrementally chip away at the foundations of these problems until you can wrestle with the more advanced concepts. If you want to try everything Brilliant has to offer for free for a full 30 days, visit brilliant.org/clockwork or scan the QR code on screen right now. Or you know what, you can click on the link in the description. You'll also get 20% off of an annual premium subscription. Give them a shot, you genuinely won't be disappointed. But for now, let's get back into the video and discuss our next paper, talking about a critical breakthrough that'll help us understand how meiosis works. And some background first. One of the coolest things that happens in our long, stumbling road to scientific progress is sometimes separate teams solve the same problem at the same time. One of the best examples of this is how the science of chemistry advanced enough that 3 independent researchers all could reasonably claim they discovered oxygen as a separate gas across the mid-1770s. Communication is way faster now, and there's enough networking across all of academia that sometimes separate research teams announce big breakthroughs by coordinating the publication of their research. And we got a big one of those a little earlier this month when 3, count 'em, 3 separate papers all hit Nature at the same time, describing a massive unlock for our understanding of meiosis. Teams from UC Louvain in Belgium, the Sloan Kettering Cancer Research Center, and the Shanghai Institute for Biochemistry and Cell Biology all released their take on a breakthrough 25 years in the making. Specifically, they all developed an in vitro model that creates DNA double-strand breaks using a key protein from meiosis, SPO11. And if your jaw isn't on the floor already, that's okay, I got you. Let's break this one down a little, starting from the top. So let's focus on meiosis first. Your body is made up of trillions of cells that, depending on the cell type, are basically always growing and dividing via a process called mitosis. At the broadest possible level, a cell undergoing mitosis replicates an identical copy of its DNA, then splits in half, sending a complete, full copy of your genome to each daughter cell. Meiosis is a very specific and incredibly important offshoot of mitosis that only happens in sex cells, like, egg cells in people with ovaries and sperm cells in folks with testes. Your cells each have 46 chromosomes that are actually 23 pairs of homologous chromosomes. One half of these pairs comes from your mom, and the other comes from your dad. You don't necessarily express genetic information from both of your parents, but you still hold on to 2 complete genomes from both of them, which is crucial for a lot of reasons, but let's stay focused. In meiosis, you start out with both of those pairs of chromosomes, and then those cells go through 2 rounds of cell division, leaving you with 4 cells with 1 chromosome copy each. That way, when sperm and egg get to know each other well enough, they'll fuse and become a cell with 2 copies of each chromosome again. Nice. But here's the superpower behind meiosis. At the very start of the process, cells undergoing meiosis will stick the matching chromosomes from your mom and dad next to each other. And like I said before, these are called homologous chromosomes. These chromosomes code for the same traits, but if your dad has blue eyes while your mom has brown eyes, they'll each contain different information, if that makes sense. Once lined up, your cells kick off something called homologous recombination, where these chromosomes will straight up swap information after a given point. This basically allows eukaryotes like us to reshuffle the genetic deck we're playing with each generation, ensuring genetic diversity while also creating a mechanism that allows harmful mutations to be filtered out before they become permanent additions to our genome. In order to pull off this gene swap, our meiotic cells have to take an incredible risk. They purposefully initiate the worst kind of DNA damage you can do, making something called a double-strand break. This basically means that both sides of a DNA molecule have been severed, which can be catastrophic if they aren't stitched back together, basically immediately. Which is going to finally get us back to the point of this paper. There is actually a protein in sex cells that purposefully does the work of cutting DNA. This worker is called SPO11. Here's a simulated look at this worker with its partner in cutting Top6BL. Nice. Okay, cool. That's half the context covered. But this half of the process honestly isn't even that exciting, because we've known that SPO11 does the DNA cutting work during meiosis for like 25 years. In fact, scientists have isolated a whole basket of proteins that help out during this high-risk DNA surgery, and honestly, academia has a pretty solid idea of how this all goes down. However, they've only been able to get SPO11 to actually do the work of cutting DNA in fully complex living cells. When you're studying a mechanism in the life sciences, there's two main ways to tackle it. You can study a system in vivo, in living cells, or you can try to study it in vitro. This is figuring out a way to engineer a more controlled environment in which you can study this mechanism. If I had to put an oversimple tagline on it, in vivo studies happen in cells, while in vitro studies happen in petri dishes, or like any other super controlled biological medium. While in vivo studies are invaluable for verifying biochemical studies and understanding sort of like the big picture in biology, they can be pretty difficult and cumbersome to use, especially if you have potentially hundreds or even thousands of different little variables you need to isolate to fully understand a mechanism. And so again, we've known that SPO11 somehow cuts DNA, because cells that have lost the ability to make SPO11 can't cut their DNA during meiosis anymore. But the how here is way more important and sometimes way harder to study. So it's a huge deal when researchers can build a stable, repeatable in vitro model of, any biochemical mechanism. And it's an even huger deal when 3 teams, which have actually collaborated a little in the past, independently come up with 3 different in vitro models that all verifiably work. Putting a button on it, our toolset for studying meiosis has just erupted practically overnight. What can we actually take away from these studies? Am I just excited that a bunch of dorks invented some fancy new kind of petri dish? Am I reading Nature papers the same way I read product pages at Hologram Electronics or Teenage Engineering, irrationally salivating over the gorgeous, overpowered specs of gadgets that won't really improve my workflow while also being ludicrously outside my price range? No, not at all. Confirming that SPO11 can work in vitro also immediately verifies some key details about how this mechanism works. The first clue is the exact variant of SPO11 that these teams used. While I make a big deal out of trying to animate the biochemistry that happens in people, mostly as a flex to be honest, most of the truly valuable information we have about molecular biology comes from model organisms. Not to freak you out or anything, but as long as we're studying another eukaryote, we can get within a few percentage points of understanding pretty precisely how a system works in our bodies. You would be shocked at how biochemically identical you are to baker's yeast, y'all. But that's a little bit of an oversimplification. And yeast usually is a solid go-to model organism for molecular biology. And in fact, 2 of the teams involved with this in vitro release actually collabed a few years back to unlock some critical insights about exactly which proteins help bring SPO11 to the homologous recombination party using yeast cells. But for what seems to be lots of little reasons, purified yeast proteins and meiotic machinery from other model organisms just cannot get the job done outside of a fully cellular context. So what was the secret to building a SPO11 model that actually works? It's all about finding the critter with proteins that actually work in vitro. The big unlock here was using purified mouse proteins. When it comes to effort, mice are definitely several rungs up the difficulty ladder in terms of how challenging it is to purify enough specific proteins to actually work with in order to do an actual biological study. A lot more can go wrong, and you can lose a lot more time if a member of your team makes one small mistake along the way and botches your results. But it also comes with some solid rewards, as mammalian systems are way closer to ours than, yeast. And more importantly, purified mouse SPO11 is capable of slicing DNA even when forced to work in vitro. The cool thing about this coordinated release is that all 3 teams took different routes to making slightly different in vitro conditions for SPO11. And so here's a model of SPO11 and its helper top6BL binding to DNA that the Sloan Kettering team was cool enough to include with their paper. Since this in vitro system is so fresh, there aren't any X-ray or cryo-EM maps of these proteins working together yet, so this model was generated by the Sloan Kettering team using AlphaFold 3. And all of the motion you're about to see is simulated by me, so take it with a big old grain of salt until new papers come out that better illustrate this mechanism. So let's take a look at a DNA strand that needs to be cut. The big thing that 2 of these papers demonstrated is that SPO11 and top6BL work together to cut DNA. More importantly, 2 copies of SPO and top are needed to pull this off. Enzymes like SPO11 have active sites where they actually do the chemical work they're supposed to do, but sometimes 1 copy of an enzyme is only 1/2 of a complete active site. That's probably what's going on with SPO11 here, maybe. It's almost like trying to cut a string with only 1/2 of a scissor. SPO11 can't make this deep DNA cut until 2 copies of it join together like this, creating a full active site. And you know what, while we're this zoomed in, let's kind of change the way we're illustrating the actual atoms here, because all 3 of these papers work together to demonstrate that this specific tyrosine amino acid in SPO11 and this little cluster surrounding a magnesium atom are the essential components of the DNA cutting action. Mutant versions of SPO11 without this tyrosine or magnesium either didn't cut DNA at all or only made it through one of the strands. And zooming out here, the UC Louvain team managed to show that in the right circumstances, just 2 SPO11 proteins can make the double-stranded DNA breaks on their own, which might open the door to an even simpler model for future study. These breakthroughs alone already are shifting the foundation of how we understand meiosis, because even these successful models show that SPO11 has a low affinity for dimerizing into the full pair of DNA scissors. That might mean that regulatory systems in sex cells have to work extra hard just to get these double-strand breaks to happen, which I mean is kind of really important considering that doing these double-strand breaks can be a huge gamble. And that helps give us a sense of one of the many critical questions that scientists are trying to answer about the molecular foundations of meiosis. Again, we now have a much clearer picture of how these double-strand breaks are made. But the big question now is why sex cells pick certain chromosomal regions over others for these double-strand breaks to kick off the crossing over and homologous recombination party. If you managed to catch my last video about eukaryotic transcription, you'll know all about how it's the regulators of a cellular mechanism that really run the show. And having the speed and versatility of an in vitro model like this one is just so huge for unraveling the regulatory secrets that power meiosis. All science compounds towards moments of revelation. Molecular biology has only really been a discipline for a little over 70 years, and researchers poking and prodding at the chemical constituents of our cells across that time period have utterly transformed our understanding of ourselves and our emergent place in this universe. An in vitro model like this simply gives the opportunity for more data gathered more specifically in a faster way. It's about to be a really interesting time to be studying this small facet of the cell cycle. But of course, it's not just understanding the foundation of our molecular diversity that's at play here. There's actually something much bigger that makes having this model important. Yes, SPO11 and its DNA slicing friends usually are only transcribed in sex cells like eggs and sperm. But you know what? There's actually a second cell type that activates SPO11 for reasons we don't fully understand. Right here, right now, do you think you could take a guess at which cell type? It's kind of a curveball, but if you're plugged in enough, you can take a look back at the 3 institutes that produced these papers and make a solid guess. Yep, that's right. It's cancer cells. For whatever reason, there are a lot of different cancer types where SPO11 gets activated for whatever reason. Maybe SPO is reverting to its ancient archaeal purpose and being used as a tool for chromatin remodeling and stability when cell division gets completely out of control. And now that we can get these scissors to cut in vitro, well heck, we can make a big push towards understanding exactly why. And dang, I can't believe this video is this long already. These are definitely my 2 favorite papers that came out in February, but there's so much more amazing research to cover. I didn't even get a chance to show you this incredible new map that shows how a small part of tarantula venom can be used to help model new classes of painkillers. But if I do a good enough job that you want me to continue making these kinds of quick and dirty videos that explore the surface level of breaking news in the life sciences, the one thing you'll start to see is just how fast things are becoming as AI models and gene editing techniques and bioinformatics and so many of these new tools in biology are becoming robust enough for wide adoption across the industry. It's so wild to me that this channel is starting to have success at the exact same moment that progress in biochemistry genuinely feels like it's starting to accelerate exponentially. So many incredible new findings are getting reported every day. It's almost impossible to keep up with. I genuinely love taking the time to explore the fundamentals of molecular biology with you in extreme detail, but it is just so exciting to try to keep up with the pace of discovery as researchers industry-wide slam down on the accelerator and we get hit by an avalanche of new and incredible papers in Nature and Science and Cell every month. And I mean, I can't make a video like this without saying it. The only thing that dampens my excitement for all this is watching unelected weirdos systematically dismantle the scientific edge my country has had for the last 70 years for effectively nothing but pocket change and extraordinarily lame memes. But weak stuff like that doesn't last. The full body of incremental scientific discovery is the only truly four-dimensional object produced by our species. Or any species that we know about, really. We're all right here, still standing on the shoulders of giants who came before us. It only matters that we can progress at all. And not to cope too hard, but the community we're building here helps give me hope that, while damaging, the disruptions here will be temporary. If we can refine our way to a more even-keeled style of reporting on all of these incredible new advancements in the life sciences, without falling to sensationalism, then that can be the small contribution we make to a more robust and well-defended scientific establishment. With all of the noise surrounding us and all the frantic pace of discovery and debate that's happening right now, don't ever forget, boring is beautiful. Sure, it took us half an hour just to walk through 2 small discoveries in the life sciences, but I hope the effort here is enough to help you slow down and try to take a longer look at all the quote-unquote small moments of revelation hitting the wire every day. The rewards of careful consideration are so rich, even if the contextual work may seem dull. And frankly, I don't even know, maybe we didn't even come close to hitting the target today, but I'm excited to build towards something better with you. But either way, that's all we have time to cover. I really hope I can encourage you to go into the description and check out the actual papers themselves. I'm only sharing ones that are open access, or at least ones that were open access at the time of publication. Reading cutting-edge research like this is genuinely difficult, but honestly, that makes it even more rewarding for me than just kind of getting spoon-fed by textbooks. I really hope you'll check these papers out, but at the end of the day, all I can really say is I really appreciate your time. Thank you so much for watching. And hey, as always, I can't thank you enough for getting to the end of the video here. If you haven't already, I'd really appreciate if you could take a sec to subscribe to this channel and check us out over at watchclockwork.com. The Clockwork newsletter is now going to be a monthly enterprise while I continue to get YouTube production more under control, but the goal here is to increase posting frequency as production becomes easier. And once again, thanks to our partners at Brilliant. Make sure you check out brilliant.org/clockwork to support the channel. And I know I literally just put this video out and you're literally listening to the end of it, but I want you to know that I'm changing this format a lot moving forward already. I was really hoping that making a grab bag of a few scientific papers would be fast enough to produce, but if you look carefully, this video was supposed to come out in February and it's about to miss March, so we're completely off target here. Moving forward, I'm going to do a bunch of shorter, much punchier videos to help me get into a more efficient production groove. I'm still building toward our next big release, which is going to be Chapter 2 of the Central Dogma about translation, but I need to give that one time to marinate properly while still serving the algorithm gods. After the Central Dogma, we're finally going to get into Season 3 of Clockwork, which is all about metabolism and sort of like the, your actual chemical foundations. And after that, we'll finally get into the good stuff for me, which is going to be immunology. But we still need to do all of our foundational work first. And sometime in the next few weeks, I'll have a more comprehensive plan posted to my Patreon that I'll be sure to link somewhere somewhere here in YouTube as well, so you get an idea of like what the roadmap is here. Either way, if we continue covering quote-unquote breaking news in biochem, I'll keep it to one paper at a time. This is the second time in 6 months where I probably should have just bit the bullet and should have just split a project into 2 separate videos. Oh well, we'll increment toward a better system or whatever. And if you want to be a part of that incremental process, once again, I'd also really appreciate you checking us out over at Patreon. Every little bit of support helps, especially since the channel is now cash flow positive, and we can start thinking about expanding out our production horizons. There's already some really great plans I have in terms of how to make our videos both better and faster. It's just a matter of making sure that the money lines up. Regardless, I really appreciate your time. It means everything to me that you stuck it out all the way to the end here. And as always, I like to leave you with peace, love, and palindromic repeats. Everyone be well. Thank you so much.